promote this kind of startups as well. There are several mechanisms. Either they give out the funding, give out the loan with the no interest, or and NASA also share the technology with the smaller license fee. So there are several things, but of course the startup itself, and also there are some other like investment day that they can do the pitching, and like kind of have the competition, and before the competition they also teach you how to pitch and do several other things. So there are plenty of activities that help to promote that. But then I think it's, in the end of the day, I think it's important to look at the technologies, if it's really scalable, if it's really commercially available. And I think our role as the researcher are very important. And I think we need to evaluate our own technology very well and work closely to the commercially, I you know commercially, I don't know, oriented people, so they know what. I think it's similar to what Dr. Hidayat just mentioned, that need to work closely with the industry to know their minds, to know what they need, and then technology and this connected. But of course, the platform government need to support, but as a researcher, you need to have a very good technology and um commercial with the commercial potential, I think. Sorry, I might not answer very quickly, but this is my idea. Yes.
Yeah. Thank you very
Thank you.
much, Dr. Lily. Yeah, we also in BRIN, we try to work close together with industry. We open the facilities, research facility for all of the people. But it's, as you mentioned, it's still really difficult to build the startup companies on a bio-based product. Yeah, thank you very much.
Thank you. I still want to add that still, even about the startup, the coordinator is important. Sometimes you need person who understand both products and both business, and to help you with that, to follow through, to make sure that it's go to the end, like, and it succeed this goal. Otherwise, like, sometimes you will be in the situation where the researcher was told that, oh, okay, if you want to do this, you have to see this person, and then when you finish this, you have to see another person, and then you go to another person. It's like it never ends, and then the researchers just give up, because they said it's better to just go back and publish, right? So I think it's important to curate, or you know develop those coordinators. And I think I have to go back to the course that I just mentioned to you. This is for the researchers, or for the scientists who want to be, in the end, you can coordinate everything as well. So this ASEAN researcher program, I encourage you all to look at it, and maybe encourage your young scientists in your lab to apply, because there is an age limit. It's like 35, so not more than 35. So please encourage your younger generation to look into this course, and maybe you can be trained to be, not just a researcher, but you can be coordinating many important projects as well. Thank you.
Thank you very much.
Yeah. Thank you very much, Dr. Lily. So actually the keyword is one person who can facilitate, right? Both of the science and also management and business. Okay, have a great day, Dr. Lily.
Thank you so much. Thank you. Have a great day, everyone.
So we would like to continue for our plenary session. Okay. Our first plenary session is with the topic Waste Biorefinery toward a sustainable circular bioeconomy, a solution to global issues. He's the president of International Bioprocess Association Malaysia, Director, Sustainable Food Processing Research Center, University of Nottingham, Malaysia, Co-Director, Future Food Malaysia, Professor of Biochemical Engineering, Department of Chemical and Environmental Engineering, Faculty of Science and Engineering, University of Nottingham, Malaysia. His research interest is in biochemical engineering, bioprocess engineering, microalgae technology, bioenergy, and biorefinery. He also received honor and award from Tan Sri Emeritus Professor Agustin Ong International Special Award on 2021 and on 2020 APEC Science Prize for Innovation, Research and Education Aspire Malaysia Award. So ladies and gentlemen, please welcome our plenary session speaker, Professor Paul Lok Show.
All right, everybody, good morning. All right, please allow me to share my screen right now. Right, this is the screen. Okay, thank you so much. Okay, so let me start with my presentation today. I given 20 minutes, so I hope that you will bear with me 20 minutes. I will try my best to finish it within the time given. So this is my first slide. This is not my last slide. Yeah, I always start with thank you. I want to thank the 9th International Symposium for Innovation Bioprocess Indonesia on Biotechnology and Bioengineering 2022. So my name is Shopal, can call me Paulo. I'm from Nottingham, Malaysia. So next slide, please. Right, so just a quick introduction about myself. I believe just how Professor Diana have mentioned about it. Yeah, so just a little bit talk about my myself. Actually, currently I'm a technology advisor under this high value, high tech agriculture, Ministry of Agriculture in Malaysia. So in this, actually I'm the advisor whenever there is a new project, new proposal, and also new finding from this Malaysia agriculture. So I'm working on this. Also a university ranking advisor under this high Mocha University. Also a founder and president of International Bioprocess Society in Malaysia. Director of Food Processing Research Center, Nottingham, Malaysia, as well as co-director of Future Food Malaysia, because excellent. So I just got promoted to become a full professor in biochemical engineering 2019. It's a very young professor, I can say. I also visiting professor in this Wenzhou University. I got my PhD in bioprocess engineering in 2 years, as well as right after my PhD, I further another degree we call PGCG, currently a fellow of higher education academic, chartered chemical engineer, which is ICAME, as well as next please. As well as fellow of Industry of Chemical Engineering. So I also Asia Chinese and professional engineer, professional technology, and corporate member of IEM. Right. So just a quick introduction about myself. As mentioned just now, our research actually covered in three areas. The first one is a bioprocess from upstream to downstream. Second is separation and purification engineering. The third one is more toward biorefinery. We call it a microalgae biorefinery. So in these three research area, I will combine it. I will share with you how we work together in the teams, yeah, in the big umbrella. So let's move to the next slide, please. So today's topic, I will share with you what is the waste biorefinery towards a sustainable circular bioeconomy, a solution to global issue. Yeah, because what I'm going to highlight today, actually we divide into 3 topics. The first topic, which is I will share with you what is microalgae first. So after microalgae, I will share with you what is biorefinery, because I believe biorefinery is very important in this circular economy. Then I will share with what is artificial intelligence. Then I will share with you the latest technology developed by our team. We call it liquid biophasing system. All right, next slide, please. Right, so first of all, what is algae? Because everybody heard about algae. Actually, algae, you can get it from a lot of water, seawater, fresh water, as well as the wastewater. Right, so next slide, please. Right, so you can see algae, we have a lot of products, yeah, a lot of, it's actually, okay, we can say it's a plant-based, yeah, you can synthesize a lot of products. So just next please. So you can see algae actually is a plant plant which can grow rapidly, I can say, can grow very rapidly, can grow multiple times within a day, grow with the help of carbon dioxide, which is free of charge, because what we want is reduce carbon dioxide and with free sunlight as well as nutrient-rich water. Nutrient-rich water can be wastewater, can be seawater, and can be fresh water as well. The range of this actually can form unicellular to multicellular. Unicellular meaning that single cell, multicellular multiple cell, resize from 0.5 micrometer, which we consider microalgae, which we already use. We cannot see using our naked eyes, we only can see by our microscope. While like a macroalgae, which is big algae, which can long as big as 100 meters, we call macroalgae. In layman term, we call seaweed. Right, next slide, please. Right, so this is the composition of microalgae. So you can see we have required sunlight, which is free of charge. We have available better of sunlight, yeah, especially in the area Indonesia as well as in Malaysia. Water can be wastewater, fresh water, and seawater. Inorganic carbon, which is carbon dioxide, and also NPK, which is nitrogen, phosphorus, and potassium. So the main composition in microalgae divide into three. The first one is a carbohydrate, which can have a lot of byproduct. As one example, we have bioethanol, bioprotanol, bioplastic, which is a product can get from microalgae. One protein is the main protein. I believe you heard about spirulina, right? Spirulina actually is a microalgae, which can produce a lot of protein. Can make in animal feed, can make in the biofertilizer, human nutrients. While for lipid, you can make a lot of biofuel, right? Example, biodiesel and nutraceuticals. Right, so next please. So we cannot forget also that is a very minor things compound available in this microalgae. Example, vitamin, mineral, pigment, and also polyunsaturated fatty acids. So this actually very important. Why I say so, because all this, right, you cannot get in most of the animal, the food that we eat, but you can get it from microalgae. Right, so next slide, please. Right, this is a short video. I'm not sure you're able to see it or not. Try to play it. You're able to play the short video here. I think it's a sour actually, yeah. There's the audio. Can you play the audio as well? Maybe let me share my screen.
10,000 million by 2050?
Yes,
How
correct.
can we produce 60% more food than today in just 3 decades, when the climate change and eroded soils are greatly affecting agricultural productivities? With this critical scenario, we need to find innovative solutions to help us tackle the problems. In this context, microalgae have real potential to contribute towards the sustainable development. But what do you really know about microalgae? Microalgae are microorganisms that first appeared on Earth around 3,000 millions of years ago and created the atmosphere as we know it today. They are the first organisms that performed oxygenic photosynthesis and are therefore the origin of all known plant-based life. They are the reason why we have oxygen on Earth. And still today, microalgae living in the oceans provide us with more than half of the oxygen we have. Also known as phytoplankton, they are the first link in the aquatic food chain. However, we have not paid much attention to them until now, because we now know that they have great potential to improve our lives in many ways. There are more than 60,000 different species, all of them with different biochemical composition and properties, hence with many different potential applications. Furthermore, they are highly productive, even allowing a daily harvest. They don't compete with conventional agriculture since they don't need fertile lands to be grown. And they are an inexhaustible source of biochemical richness, including proteins, omega-3 fatty acids, carbohydrates, carotenoids, polysaccharides, vitamins or phytohormones, among many others. Thanks to this composition and their biodiversity, they are a raw material with immense potential for application in many different sectors. Microalgae can be a key part of the solution because they are a source of clean, sustainable, and inexhaustible biofuels. Because they are one of the most efficient CO2 biofixation systems in the world. Because they are a novel food source cataloged as the food of the millennium by the FAO of the United Nations. But more importantly, microalgae-derived products are already playing a key role in considerably increasing productive yields in agriculture, aquaculture, and animal production. For all these reasons, we not only owe microalgae life as we know it, but also the improvement of our lives in the coming years. They are our origin, but they are also our future.
So this is a video about, actually this video is talking about the whole summary. What is microalgae? What is the composition? Why we need microalgae? So as per the video mentioned that microalgae actually is our origin, yeah, because our human exist in the world, there's microalgae really. And microalgae also our future. Why? Because microalgae can be a future food, can be a future fuel as well. Right, so let me proceed to the second topic that I will share with you, which is why microalgae biorefinery. So you can see in microrefinery, as I mentioned just now, right, we have lipid, protein, carbohydrate, and other pigment. We call it algae biomass. Because this is microalgae, we have a plant-based, we have very thick cell wall. We need to undergo a pre-treatment to break the cell wall. So what is the pre-treatment that we can apply it? We can sonification, microwave, enzymatic, hydrothermal, pulse electric, and also thermal shock. So this is a pre-treatment to break the cell wall. After breaking the cell wall, right, so you can see a carbohydrate, lipid, and protein. Undergo fermentation, you will see a lactic acid. So for lipid undergo transfiguration, you have produced glycerol and this cell. So you can see some lactic acid as well. So for protein, you can undergo some process. You will produce fertilizer, supplement, and also food product. So all these processes, right, you will involve on wastewater. So for the wastewater, yeah, you can reuse it, recycle it, and for the cultivation of microalgae. So the whole process, actually, we call it microalgae biorefinery. So you can see it actually is a circular economy as well as zero waste discharge from the system. Right, next slide, please. Right, so next topic I will share with you is why we want to put artificial intelligence in the system. Because we sometimes we need to think in the positive way. Even we can see pandemic give a big impact, but pandemic also give us some benefit, which is we have no choice, cannot go to lab. So we have to learn something from home. So that's why in my team, right, we develop artificial intelligence in this microalgae biorefinery. Next slide, please. So I will share with you what is artificial intelligence. So actually, just a quick preview of artificial intelligence, actually referring to a simulation of human intelligence. So actually, we are going to train a machine that are programmed to think like a human and mimic their actions. So whatever that you think, whatever that you want to do, you can do the coding, programming, and do the simulation of it. So we call it artificial intelligence. So our team will develop this artificial intelligence in this photobioreactor, as well as to using mobile phone to do the real-time monitoring of the cultivation process, extraction. So next slide, please. So just to recap for you, yeah, what is the first industrial revolution? We focus on steam engine. While next please, second revolution, we focus on electric motorized assembly line. That's where you can see a conveyor belt, a lot of conveyor belt in the industry. Third one, actually, we focus on roboting with a robot. This way you can see a robotic arm. For industry revolution, with a lot of people talking about production of system that learn and adapt the predictions. So this is we call our fourth industry. So after that, we combine everything together. We come up with a system we call a liquid biorefinery system. Actually, it's a new biorefinery techniques. So let me share with what is liquid biorefinery system. Right, next slide, please. So liquid biorefinery actually is a liquid partitioning method of two liquid phases. So we involve a liquid system in the system. Actually, we involve two liquid together. So in our team, we successfully recover as separate magnetic compounds. So some example I can share with color pigment, protein, carbohydrate, lipid, vitamin, mineral, enzyme, nutrient, aroma, heavy metal, active. So whatever that you mentioned, that we name it, so we can use this liquid biorefinery to do the separations. So in the next slide, I will share with you how to do this liquid biorefinery system. So in order to prepare liquid biorefinery, it's actually very simple, just mixing and shaking. Just how you make coffee. I believe in coffee, like coffee, creamer, sugar, and also hot water. So in this liquid biorefinery, actually very simple. You just mix everything. There is another gradient force. In this liquid biorefinery, there is two main factors affecting the system. The first one is the phase forming component. Just like make coffee, you need robot Arabica coffee bean or Robusta coffee bean. So you can see you can add a self-destructive agent as separation preparation agent. So the first step actually you put all this together, then you can see next step is you have the phase separate. It can be a salt, sugar, iron liquid, all this. So you can see all the particular right potential at the top. This we add as a separation and purification agent. Right, next slide please. So the next thing I want to share with you are the smart extraction technology in liquid biopsy system. So what is smart extraction technology? Actually, smart things meaning that we implement the artificial intelligence system. So extraction technology actually combining upstream and downstream together. So how work in? Actually, one phase you can see is a phase forming component. Phase forming component can be iron liquid, can be polymer, can be salt, can be sugar, plus a feed which is neutron, medium, and microalgae biomass. We do the normal cultivation or process. After that, we heat it above the critical solution temperature. A lot of people will question why we need to heat it, because we deal with live organism. Heat will involve a lot of energy, but in that case, we don't heat too high. We only heat 40 to 50 degrees Celsius. We want to see the two phases. On top, after heating process, you can see top phase consists by product and water. You just remove the water, you get your pure product. Pure product can be lipid, protein, carbohydrate, DHA, pigment, whatever that you want to extract. Bottom phase consists of phase forming component biomass, which you can recycle. So this is how we work together. We call it our bottom phase recycling system. Of course, we put the smart extractor system. So this video we can share with you how we do the simulation of simulator of cultivation and biorefinery microalgae. Yeah. So you can see from this video, right, this is a single step extraction. So we're using alga culture separation by this. So at the same time, we cultivate the microalgae. At the same time, we do the extraction of microalgae active compound from microalgae. So you can see after the harvesting system, we heat up above the critical solution temperature. You can see two phases. Yeah, two phases. Top phase you can see are all biomass. All right. So after that, after the critical solution temperature, right? So maybe 40 to 50 degrees Celsius, you see the two phases. On the biomass, we break the cell wall and we will partitioning to the bottom phase. So on top of it, right, you can see a thin layer of oil. Bottom phase, you can see a water. And this one is a biomass used to recycle. Yeah, why we claim it as smart? Because we put some sensor and we can put a Wi-Fi. This one we can monitor using our handphone. So time to time we can see the concentration of the biomass, concentration of water, and concentration of our oil layer on top. So this is why we claim it as a smart technology. Yeah, this method we already patented and we use it in mining industry. So let's proceed with waste biorefinery toward a sustainable circular economy, a solution to global issue or not. Yeah, actually, yes, I can say we need to have a right combination at the right time in the right place. Yeah, because a lot of things happen around there. So I think this is our new book. I can just want to share with you. Actually, starting from 2019, we think that application is not enough to reach a wider audience. That's why we come up with a books. So first book we produce, we call bioprocess engineering. Second book is that we have is the liquid biopsy system that I will share with you. The third book actually we produce the prospect of Industry 5.0 in binary manufacturing. So you can have a look. I think the latest book that we produced this year, we call it Microalgae for Environmental Biotechnology. So this is the latest book that we produced. Actually, you can check the book in most of the bookstore and also Google Book already. Right, next slide, please. All right. So this is my next slide. I think this next slide is a lot of information, to share with you our website as well as to keep in touch. I believe some of you may be shy to ask questions now, so you can text me or email me anytime that you want. Right. So this is my WhatsApp, my Facebook, my Talk, and my Zalo, and my WeChat as well. So this is my website. So thank you so much.
Thank you very much, Professor Shaw, on very interesting presentation on how beneficial of biorefinery of microalgae, with the emphasis on novel purification and separation using liquid bioplastic systems or circular bioeconomy. So we will have discussion after one more plenary speaker.
Thank you.
Okay, let's continue to the second plenary session. Our speaker for the second plenary session is Dr. Tony Hadibarata. He's an associate professor from Faculty of Engineering and Science, Curtin University, Malaysia, and also a young professor on Center for Coastal Disaster Mitigation and Rehabilitation Studies, Faculty of Fisheries and Marine Science, University of Diponegoro, Indonesia. He obtained PhD and master degree in environmental chemistry and microbiology from Ehime University, Japan, and Bachelor of Science from University of Mula Warman, Indonesia. His research interests in environmental chemistry, environmental microbiology, environmental engineering, bioremediation of toxic pollutants, water and wastewater treatment, and also in microbial enzyme with H index 37. Please welcome our plenary speaker, Dr. Tony Hadibarata.
Very good morning. Hello, can you hear me? Hello?
Yeah.
Okay. Yeah. Okay, so very good mornings, everyone. So today I would like to share my research about recent advance on biodegradations and bioremediations of emerging pollutants. Next slide, please. Okay. This all about waters. In Malaysia, the situation is 90% of water supply in Malaysia is came from the rivers. Unfortunately, the pollutions of river is increasing. So until now, I have the data until 2019, still 10 polluted rivers, as well as 49 slightly polluted rivers. So the pollutions is came from the point source, from the sewage treatment plants, manufacturing, agro-based industry, as well as from non-point source from the agricultural activity, plantation, and surface runoff. Next, please. Okay. The problems in the wastewater treatment system in Malaysia is only effective for the conventional pollutant, as well as the BOD, COD, THS, and so on and so on. But there is no parameters effective for the emerging contaminants removals, as we seen from the tables. Most of them still showing low removings for the emerging contaminant. So it is very important to create the effective treatments, especially for the emerging contaminants. Next, please. Okay, the emerging contaminant is the pollutant. Actually, the pollutants has been detected in the water bodies. But the problem is there is no regulations, no regulation, no policy, or no environmental laws to manage or to arrange about this kind of pollutants. So in this study, I will focusing on the pH, polycyclic aromatic hydrocarbons, as well as the microplastics. If you check the publication of these two emerging contaminants, increasing year by year. Next, please. Okay, so we start from the biodiversity in Southeast Asia. As we know that Southeast Asia, or ASEAN regions, is having a highest biodiversity, including the non-wood product. We are focusing later in the non-wood products. So until 2015, 206.5 million hectares of forest cover Southeast Asia, but currently the problem is the deforestation, increasing year by year. Here the data, you can see the data that increasing of deforestation from 2001 until 2016, as well as the increasing of the production, productions forest. Next, please. Okay, this is the Sarawak. Sarawak is 37% of Malaysian regions, consisting 800,000 of protected areas, national parks, wildlife, nature reserve, and currently Sarawak has 15 national parks and open to visitor as the tourist destination in Sarawak. Okay, so here we focusing on the mushroom, the mushrooms that we want to utilize. Next, please. Okay, this is the utilization of mushrooms in biotechnology for biopulpings, for the brewings and the reproduction, as well as the fermentations. But in this study, I would like to focus on the environmental management, utilize some enzymes extracted from the mushrooms, such as the lacase, the phenol-oxidase, lipase, peroxidase, and phytase. Next, please. Okay, maybe this is everybody already know, but the type of wood decay fungi. We have 3 types of fungi: white roots, brown root, and soft root. That why we use this fungi, because the similarity of the lignin, because the fungi, the component of woods can be degraded by the fungi, and we estimate that if the fungi can degrade the wood component, especially lignins, so the fungi can degrade the organic pollutants, especially the benzene rings pollutants. Next, please. There is 6 categories for the enzymes, but for the environmental remediations, only 3 playing roles: oxidoreductase, transferase, and hydrolase. So in this study, I will focusing on the oxidoreductase as well as hydrolase. Here, because these 2 enzymes is more effective for the pH as well as the microplastic. Next, please. Okay, the 3 ligninolytic enzymes is consisting 3 types: the lignin peroxidase, lacase, and manganese peroxidase. Every enzymes has characteristic. For the lignins, need the peroxidase to degrades or to substrates or to oxidize the pollutants, and for lacase need the oxygens, and for the manganese peroxidase similar with the lignins but need the ionic manganese ions for the process. Next, please. And the lacase is one of the powerful enzymes due to the effectivity for degradations various pollutants, including the emerging contaminants. So there is 2 types of oxidations. First is direct without any addition of mediators, and the second is indirect with the additions of mediators. Of course, the 2 types oxidations can degrade the pollutants, but mediators increasing the times and the process for transformations. Next, please. Okay, we are starting from the pH. We start from the beginnings, the screenings of the mushroom. So I collect egg hundreds for Basidiomycetes from several national park in Sarawak, Piasau, Lambir, Nia, Simalajo, and Bako. And these various forests, representing the hill mountainous tropical forest, the wetland forest, as well as the swamp forest. So the fungi is isolated using the agar mediums, and we check the effectivity or the resistances in 4 indicators. We use the poly R478, Remazole brilliant blue R, guaiacol, as well as the siningal dasin. You can see that structure of this indicator is similar with the structure of the emerging contaminant. And then we screen based on the growing as well as the decolorization, and we identify the names of the fungi. Next, please. Okay, this is some of the pictures that collected from the forest. We collect 459 species during the wet seasons and 345 species during the dry season. So 62% is belong to the saprophytic fungi, and as well as the edible 38%, as well as the inedible. Next, please. So the identifications of the fungi was conducted by microscopic, by observing, touching, smelling, testing, microscopics by the measure the size and shape of the spore as well as the arrangement of the spores, and the finals, the molecular identifications by sequencing of PCR amplified part of the 18S RNA with the universal primer. Next, please. And the degradations process conducted like this. Yeah, this is the flow. Yeah, the flow of the experiments. First, the media preparations, and then culturing, incubation for several days, extraction using several solvent combination. Yeah, including the separations using the column chromatography. And for the enzymes productions was identified or with the UV fish spectrophotometers with some substrates. For the detection of the metabolites is checking using the GCMS, yeah, GCMS the finals, but we have to check also the melting points, the UV fish as well as the TLC, yeah. And then we compare the results of the sample with the standards in the GCMS, or we can compare with the synthesis if we don't have the compounds. Also checking in the library of the GCMS. Next, please. Okay, this is the mechanisms. Yeah, the mechanisms. If the mycelia grow, they will produce the enzyme. Yeah? There is the enzymes. The enzymes will extract, yeah, to the liquid culture, and liquid culture will oxidize the pollutants, yeah, the pollutants in the liquid mediums, and convert it into the simple structure of chemical. And if the simple structure is composed, the compound will be absorbed to the fungal body and degraded in the fungal body. So we compare the sample with the standards library as well as the synthesis using the UV fish, melting point, TLC, GCMS, and FTIR. Next, please. So we can see here that most of the pH, yeah, penetrance, anthracene, and pyrines, 3 rings and 4 rings pH was degraded, yeah, was degraded more than 60%. The question is the pollutants, or are the pollutant eliminated by biosources or by biotransformations? Yeah. So we have to check letters. Next, please. Okay, here we can see that we found some metabolites, yeah, some metabolites from the penetrance, from the anthracenes, as well from the pyrene, yeah, and as well as the fungi that playing role in the degradations. We can see from the right columns. So we can conclude that the removal of the pH in the culture is conducted by the transformations process. Next, please. Okay, this is the pathway of the penetrant degradation. Basically, the most of the pH, yeah, consisting the Bay regions and K region, and these regions, yeah, this both region is most active in compared with the other regions. So we can see here that the K regions was oxidized, yeah, was oxidized by the enzymes to form depending acids, 1-hydroxy-2-nitric acids, hydroxybenzoic acid, as well as the pyrocatechol. The compounds in the brackets, between the bracket, was not identified in our culture. So this is confirming with the table previously, yeah, the table of the spectral analysis. Okay, next, please. And this is for anthracenes. Yeah, anthracenes. There is 2 role of enzyme here. The first role by lacase, yeah, to convert the anthracenes into the anthracene quinone, yeah, anthracene quinones, and finally forms the phthalic acid by the intra and extra enzymatic conversions or end-ring fissions to finally forms a benzoic acid. In the other routes, yeah, the dioxygenase and the dehydrogenase converting the anthracenes into the anthracene 1,2 diols, and finally form the benzoic acid. So unfortunately, we cannot find the both compounds between the brackets. Next, please. And this is the pyrenes, yeah, similar with the previous metabolites. So there is 2 pathways. First one is phthalic acids, because the final product is the phthalic acids, as well as the salicylic acid pathway. Yeah, so we can confirm that all pH has been transformed, yeah, by the enzymes of the fungi collected from the forest. Next, please. Okay, so the problem is, can the treatment be applied in situ? Yeah, can the fungi or the enzymes can be implemented in situ? So, of course not. Yeah, we have to to immobilize the enzyme. Yeah, immobilize the enzymes to make sure or to ensure the activity and stability still in the high performance. So there are 4 immobilization methods: cross-linking, adsorptions, covalent bindings, and entrapments or encapsulation. So each method has the advantages and limitations. And I will try to do the entrapment or encapsulation to combine the enzymes with the absorbent as well as the magnetic force. Next, please. Okay, so this is the grant, the grant from the Ministry of Higher Education about the prototype research. Yeah, so we combine the enzymes with the absorbents with the magnetic powers to produce the magnesium. We call it the magnesium. Yeah, next, please. So the magnesium consisting the enzymes, yeah, in the middle of the prototypes. The outer layer is adsorbent, yeah, from the lignolysis waste, and the magnetic core in the inside. So we can implement, yeah, the prototypes in situ, yeah, especially in the liquid or aquatic environments. So, for example, if there is a crude oil contamination, so we can spray the magnesiums there, and then we can eliminate the magnesiums using the magnetic force. Next, please. So how strong the magnesium to degrade the organic pollutants? So we can see here the magnesium only cannot be effective only in the soil, yeah, because mainly focusing on the aquatic environment. Okay, next, please. The other alternative method is using enzyme dispersion. So we combine the enzymes from the specific microbes, and then we combine with the aqueous enzymes and making formulation with the ionic liquid to perform the enzymatic dispersion. So the mechanism is similar with the magnesiums, but the shape of the prototype is liquid. Yeah, so we can spray in the aquatic environments, and then we can convert or we can eliminate the crude oil or oil spills in on the sea. Next, please. Next, we are focusing on microplastics. As we know that the consumptions of the plastic, yeah, in ASEAN, yeah, increasing year by year. But you can see here the Malaysia is highest in the ASEAN country, 16.78 kilogram per capita, yeah, per capita. Next, please. So in order to this reason, we want to check, yeah, the occurrence of the microplastics in some river in Sarawak, as well as in the commercial marine fish in some market in Miri. Yeah? So we check 3 rivers, yeah, 3 rivers which we collect the water and sediments, as well as the 5 fish from markets: skipjack tuna, mackerel, sea catfish, shrimp, bivalve. Yeah, each sample 2 kilograms per market. Okay, next, please. Yeah, this is the mechanisms how to analyze the water sample, the sediment sample, and commercial fish. In this study, we just only check the shape, size, and color of microplastic, as well as the functional groups. We didn't check until the mass concentrations, yeah, because we need the advanced equipments, and we don't have the equipments at the time. Yeah, next, please. So here the study, yeah, 3 rivers: Baram rivers, Miri rivers, and Nia rivers. Yeah, if we compare with the other rivers in ASEAN, yeah, Indonesia and Thailand, yes, quite similar. Yeah, we can compare similar. But if we check with other regions, so some rivers showing higher than the Barams, Miri, and Nia rivers. Next, please. And for the commercial marine fish, yeah, you can see that varies from the 11 items per kilograms until 20 items per kilogram. So we can compare with the other fish here. Yeah, some of them showing the high occurrence. For example, like yellowfin tuna, 5 per gram. Yeah, this is per gram, not per kilograms. So if we convert to kilogram, maybe more than 500 or 5,000. Yeah. Okay, next please. Okay, this is the situations in the Baram River. Yeah, Baram River. We can see the construction of the microplastics in the Baram rivers. Yeah, fiber, pellets, fragments, films, and foam. Fragments showing the highest. Yeah, compared with the other shape of microplastics. Next, please. For size, yeah, for size we can see here the smallest size, yeah, 300 until 1,000 micrometer, showing the highest, yeah, because maybe the degradation or the deteriorations of the microplastics happens during the process of secreting, yeah, or extracting in the river. Next, please. And for the color, you can see the blacks and blue showing the highest, yeah, for the occurrence of microplastic in Baram rivers. Next, please. And for the Miri rivers and Niha river, this is the configurations. I will not explain in detail, but yeah, this is the configurations for the size, the color, and the, yeah, okay. This is the occurrence of microplastic in fish, yeah, in fish, in commercial fish. You can see here the configuration of color, and then the fragments or types, as well as the size. Yeah, next please. This is the picture, yeah, the picture that we found from the water sediments, yeah, as well as from the commercial fish. Yeah, you can see here. Yeah. Next, please. Okay, yeah, this is the when we check the microplastics in the FTIR. So you found that the types of microplastics belong to the polyethylenes, yeah, polyethylenes, and then the polypropylenes, polyurethanes, as well as the ethylene phenyl acetates, ethylene propylene monomer, as well as the butyl bromide. So we check based on the stretchings of the carbon and hydrogens, as well as the bending of carbon and hydrogens bond. So basically, when we check again in the sample of the fish, mostly PE, yeah, PE is showing the highest concentrations of microplastic. Okay, next please. Okay, yeah, this is the mechanisms of screening, same with the previous one, yeah.
Dr. Tony, 1 more minute, please.
Okay, okay, yeah. Okay, so this is the mechanism similar with the previous one. So, but the indicator is changing with the polyethylene, yeah, polyethylene. So we can find, or we found that the aspergillus integer is the strongest, yeah, showing 92% in 35 days, yeah. And when we check the enzymes, showing the cutinases, hydrolase, and esterase is showing the highest performance for the enzyme. Next, please. Okay, this is the mechanisms of degradation. Yeah, the degradations, enzymes is attached to the surface of the polymers. Yeah, and then the depolymerization happens, biofragmentation happens, become to monomers, and then the monomer will be absorbed to the cell. Yeah, the internal cell, and internal cell will convert the monomers into the energy and the CO2, H2O, and or CH4 if the mechanisms of anaerobic. Next, please. Yeah. When we compare with other, when we compare with the other microbes, yeah, aspergillus is showing the highest, yeah. But the problem is this is only for PE. We don't know for PP, PS, and PPG, yeah, as well as the metabolite of microplastics and the impact of aquaculture on microplastic contamination. So for the next, we will try to investigate these problems. Next, please. Next, please. Okay, yeah, this is the conclusion. Yeah, the conclusion that enzymes, yeah, can make an effective, sustainable strategy for the emerging contaminants degradation. I think that's all for the my presentation because the time is over. Yeah, maybe you can show the journal. Yeah, the journal maybe for the, if audience want to download the paper. This is the paper, our paper for the presentation, as well as the acknowledgement from the various grant from Malaysian government. Thank you very much.
Thank you, Dr. Tony, for great presentation. So we will have discussion. I would like to invite Professor Show. So for those of you who have questions, you may use chat, or you can raise your hand also. So this is there's a question for Professor Show from Ibu Penny Lestari, BRIN. One is, are all microalgae species beneficial, or is there also dangerous? And the second question is, how is the quality assurance of this microalgae related to purity?
Right, thank you for the questions. Yes, yes, you're right. Yeah, some microalgae, we cannot deny that some microalgae is harmful, some microalgae is not good. Yeah, actually, microalgae can divide into two categories. The first one is beneficial algae, which is can have a lot of product. Yeah, can give us some protein, carbohydrate, lipid. All this we call beneficial algae. While harmful algae maybe has some toxicity, may some ingredient that is not good for a human being. We call harmful algae. Yeah, because that's why in a lot of cases, right, we need to select very carefully the algae that we consume, that we use. That's why we need to do the isolation, purification, all this to select the good algae. Yeah, just to share with you, actually in algae, right, if you grow too much algae, we call it algae bloom, right? It's not good, as we can consider it as a harmful algae as well. So that's why it's a good and harmful algae, right? For your second question, actually, is relating to the purity, right? So I can say, the quality of the algae is very subjective, I can say. Yeah, because as I mentioned in my presentation, need to have a good purity of algae. If not, there are so many impurity, so many like example, the material that we don't want, the chemical that we don't want contained in the microalgae biomass, which is not good for human consumption, or not good for animal as well, not good for other product as well. That's why the purity is important. Yeah, because in the traditional way we produce algae, we produce as much biomass, which we call upstream of bioprocess. So we cultivate as well as bioprocess, but in the bioprocess, right, we have a lot of ingredient purity that we don't want. That's why we need to go through a downstream processes, which is we need to do the purity extraction and also the recovery of the product from microalgae, so that we can use it in any food supplement, medical, as well as a cosmetic product. So the purity will play a very important role in the quality of microalgae. Thank you so much.
Okay, one question for Dr. Tony from Bapak Dani from BRIN. Some fungal cytochrome P450 also capable to catalyze biotransformation of PEH like oxidoreductase. However, the biotransformation product must be different because P450 cannot catalyze the formation of tyrosyl radical like oxidoreductase. Do you experience in the comparisons study of enzymatic activity of fungal oxidoreductase and for P450 against PEH? And second question is, did you identify PE degrading enzyme in Aspergillus niger? Are they PET hydroxylase?
Yes, thank you very much for the question from Dr. Dani. Basically I use here the peroxidase, yeah, because the oxidoreductase is comprises three types, yeah, like lactase, peroxidase, and the oxygenase, yeah, oxygenase divided by the mono and the deoxygenase. So I think P450, they will convert to not area of the K or B region. So basically in other that both area. So they will not making or not producing the quinone compound for the pH. Yeah. If you see my slide previously, most of the PAH is converted to the quinone, the quinones, because of the peroxidase and lactase. But for the P50, I think not making the quinones, but directly to the other pathway, maybe protocatechoic acids or the salicylic acid pathway. And for the identification of the PE degrading enzyme, we check at the beginning the cutinase, the cutinase and hydrolase, as well as the esterase, the enzymes detected in the Aspergillus niger culture, But we will try again. Is there any peroxidase or any oxidoreductase enzymes, producing by this microbe, Thank you.
Okay. I will give an opportunity to Ibu Yeni.
Thank you so much. Could you hear my voice?
Yes.
Okay, thank you so much. I would like to ask Professor Paulo.
Your institution, please.
My institution is from the National Research and Innovation Agency. So I would like to ask Professor Paulo about the production of the microalgae technique. So actually, I have an experiences of using the wastewater as a growth media for the microalgae. But as we know that sometimes the wastewater have like unbalanced nutrients that can not stimulate the high production of the microalgae, So I would like to ask you if you have any experiences of using the wastewater and how to manipulate the wastewater to improve the growth of the microalgae without involving any like a chemical fertilizer or something. And the second question is about the composition of the microalgae itself, for example, like carbohydrate, protein, or liquid, or something like that. So, for example, I have an industry of the bioenergy, and I want my microalgae having a high carbohydrate, for example. Can we manipulate, like, for example, the growth condition of the microalgae so we can have the high content of the carbohydrate of our microalgae species? Thank you so much.
Right. Thank you so much, Ibu Hile, for the questions. Right. So to answer your first question, actually, I have some experience in cultivate microalgae using wastewater. So what I'm trying to do actually in Malaysia, because Malaysia we have a lot of palm oil mill and float, we call it pomade, the wastewater from palm oil mill. So we cultivate because we have 4 ponds in the pomade. So we not dare to use the first pond because first pond is very concentrated, highly viscous, and the pH is very extreme. So what we do is we use the second, third, and fourth one to cultivate microalgae. The reason that we use third, fourth, and the fourth one, right? Because we think that the ingredient, the nutrient inside is suitable to grow microalgae. So in order to answer your question, actually, what we do in our lab first, actually we characterize the wastewater conditions, right? Whether it's suitable to cultivate certain type of microalgae. Because, like example, chlorella, spirulina, different type of microalgae, right? They need different of nutrient. So what we're going to do here, we characterize of the wastewater, which water can suitable to cultivate microalgae without doing any pre-treatment and any fertilizer involved in the wastewater. So we can direct use it. So this is something that we do in our lab, right? Yeah. To your second question, actually, what is the method? What is the idea that come up from me? Actually, you want to manipulate the concentration of the active compound in the microalgae. Very subjective, I can say, because different microalgae have different ability. Different microalgae have different strength also. Yeah, some microalgae rich in protein, some algae rich in carbohydrate, some algae rich in lipid. So I can say what we can do is we can have some microalgae strength study. So maybe we have, because currently we have our team developed a new consortium, we call it Algae Biotech Consortium. In this consortium, mainly we are collecting, I think more than 300 strengths around Asia. So maybe we can do together, we can exchange the strength. Yeah, maybe you have what strength you want, come to talk to me, then we can share some strength with you so that you can cultivate in your site. Yeah, because the strength character is important to decide what product that you want. So let's say, example, just now you want to have a rich in carbohydrate. So maybe I can provide some strain that rich in carbohydrate. So you can try to cultivate in your site so you can produce the carbohydrate from your site. So this is something that my suggestion. Of course, some strain, right, is like I say, some strain is very need to undergo certain treatment like stress condition. So you provide a stress for them, so they will provide additional product that we want, like a hemococcus, to produce azacitidine. That this is some example. So different cultivation process, different optimization process, or different condition of medium, we have different product. Right. Thank you so much.
Thank you so much.
You're welcome.
Is there any other question? Okay, I have a question for Professor Show. Does a different species of microalgae is this require specific condition of a liquid biphasic system also? And if you have a problem with contamination during simultaneous cultivation, how do you manage?
Right. Thank you, Ibu Diana, for the questions. Yeah, actually you are right. Yeah, because you for different microalgae you need different liquid biphasic system, because in this liquid biphasic system they have a lot of combination. They have salt, sugar, they have under liquid polymer, alcohol. So different product you want to extract, like example, you want to extract lipid from this microalgae, you need different of under liquid, different of salt water. Because even salt, right, they have different type of salt also. So you need different combination. Yeah, so different combination will produce different product. But you do not have to worry about the combination, because combination only happen one time, meaning that you do the optimization, everything's already, then at the same formula you can produce the same product. So just like how you make coffee, you already have correct concentration of your flavor, temperature, and the creamer amount they put inside. So you continue using that formula, that is your taste already, this is your flavor already. So I can say this is a different combination. Yeah, thank you.
Okay, thank you.
Welcome.
And for Dr. Tony, how do you manage the production costs to produce the bioactive compound to be applicable for user or maybe other stakeholder?
Okay, basically we still trying to find the cheapest way to produce the mechanism, especially for the absorbents. So we can use the absorbents from the agricultural waste. So this is cheaper than we are ordering from the supplier, or from the commercial supplier. So we create the absorbents from the agricultural waste because we have equipment here to produce the activated carbons. And for the enzymes, enzymes especially, we try to find the best fungi that produce high units of enzymes in liter or gram. So basically, if the productivity highest for enzyme productions, as well as the cheapest for the activated carbon, then we can try to lowering the cost for production of mechanisms. For the irons, the magnetic power, I think that's we cannot do anything about this, because this already settled. So we cannot adjust the price. Then we just adjusting the price for the activated carbons as well as the enzymes production. Yeah, thank you.
Okay, is there any other question? Okay, I think we can complete our discussion today. And I would like to conclude that it is important to develop a novel method or explore novel organism in the context to support sustainable development goal and strengthening circular bioeconomy and also green economy. Thank you very much for Dr. Tony and Professor Shaw. Have a great day, everyone. And I would like to let the MC to lead for the next session.
Yeah, thank you so much.
Thank you very much, Prof. Diana. Thank you, Prof.
Thank you.
So
nice to meet you, Tony. Yeah. Bye-bye.
Thank you to all speakers for the material given. Also, thank you for the moderator to lead the sessions. Following to the next agenda, we will have the parallel sessions consisting of 6 rooms with different topics. Allow me to read for each room. The first room will be bioresources and biodiversity. The second room will be nutraceutical and natural products. Room 3 will be biotechnology and bioinformatics. Room 4 will be medical sciences. Room 5, agricultural and forestry. And room 6 will be environmental and sustainability. Ladies and gentlemen, each of room will be led by moderators, and please kindly choose the room based on your interest by clicking the breakout room feature in your Zoom meeting. Thank you. While waiting for the breakout room, you can kindly take a look and also choose the room based on your interest. As you can see on the screen right now, there is 6 rooms with each topic and also led by moderators. Ladies and gentlemen, responding to your questions at the chat, how to enter the room, please guide, and also the breakout room hasn't ready yet. The official committee is still working on it. While we are waiting for the breakroom feature, you can again take a look at the based on your interest. Please kindly, thank you. Ladies and gentlemen, the breakout room is already available. You can find out by clicking more, 3 dots, and just click join breakout room. And there will be room 1 until room 6 available. Once again, ladies and gentlemen, please kindly click 3 dots at your Zoom, more, and then click join breakout room. Have you seen it? Again, ladies and gentlemen, you can click at more feature, 3 dots at your Zoom, feature more, and then there's a join breakout room feature there. You can clicking join breakout room and then join for each room available there. Thank you, Bapak Win. Yes, look at the left, at the corner, at your Zoom. There will be 3 dots more and then join breakout room. We hope you can find the feature.
Okay, thank you very much. Could you hear my voice, ladies and gentlemen?
Yes.
Okay. So welcome to this session. So I would like to moderate this discussion. And in this session, we have 6 presenters here. And there will be a 10 minutes presentation, and all the presentation will be delivered first for the 6 presentation, and then after that we can discuss about the presentation for 20 minutes. And I would like to invite Pak Danang, Pak Bowo for the presentation, and maybe Mas Dr. Imam, could you please to play the video? Pak Danang already here, right?
Yes, I'm here.
Okay, thank you very much. And also the second presenter is, wait, I will. Yes, the second presenter is Bu Hani Susanti, yeah, and the third is Bu Dini Akhnafani, and the fourth Bu Nur Absari Aji, and then continue with the Bu Wuri Handayani and also Triwira Yuwati. And now we will have presentation from Pak Danang Ambang Prabowo with the title Unwanted Heat Seekers, yeah, the potential dispersal of harmful marine microalgae via plastic garbage and debris in Indonesia waters. Okay, Pak Imam, could you please to play the video?
Hello, everyone. My name is Danang Ambang Prabowo. I'm a research scientist at the Phycology Research Group of the Research Center for Biosystematics and Evolution at BRIN. And on behalf of my colleagues, allow me to present to you our latest research findings about the potential dispersal of harmful marine microalgae via plastic garbage and debris in Indonesian waters. It is estimated that over 8 million tons of plastic reach out to the oceans, which accounts for approximately 80% of the total garbage found at different depths. Plastic pollution does not only threaten the health of the oceans, including aquatic organisms that live within, but also human life in terms of health, economy, tourism, food safety, and etc. Marine plastic garbage can also serve as attachment for various micro and microorganisms, which support their dispersal with the help of wind and ocean currents from the native environment to the new habitats. This could impose serious ecological consequences, especially when the transported species are deleterious or detrimental, such as the harmful microalgae. However, unfortunately, to the best of our knowledge, information on the potential roles of marine plastic garbage in dispersing harmful microalgae in Indonesian waters is currently lacking. So what are harmful microalgae? For some of you who are not familiar with harmful microalgae, the term is specifically used for microalgae taxa that are capable of producing massive blooms called harmful algal blooms or HABs, and or species that can cause numerous harmful impacts to the environment and organisms, including humans. There are approximately 200 well-described species of harmful microalgae, and they are divided into 7 major groups that are associated with various harmful effects, such as bloom formations, toxin productions, species that can cause physical damage to skin and gills of aquatic organisms, especially fishes, species that can release airborne toxins through aerosols, or even some species that are able to produce toxin-like compounds that can be only detected in laboratory through specific analysis. So what is the connection between plastic garbage and harmful microalgae? In this study, we hypothesized that plastic garbage can help the dispersal of microalgae, especially harmful microalgae in the marine environment. For example, let's say somebody disposed a plastic bag at the beach, and once the plastic bag reaches out to the oceans or the water, it will eventually sink to the bottom or suspended by the seagrass, where benthic or epiphytic microalgal cells, including harmful species, will be recruited onto this new substrate. Subsequently, with the help from wind, currents, or waves or tidals, the plastic containing the microalgal cells will be transported to a new habitat where they can be potentially becoming invasive alien species or becoming a source for harmful algal blooms and seafood poisonings in the new environment. So to test this hypothesis, the aim of our study was to detect whether potentially harmful microalgal species can be found in such disposed plastic materials. To do that, we collected plastic garbage and biological debris from 2 locations in northern marine region of Jakarta, namely Tanjung Pasir Beach and Tidung Island in October 2021 and in February 2022, respectively. Samples were collected randomly, and the cells were detached from the garbage materials by spraying them with filtered seawaters into 50 milliliter Falcon tubes. The cells were subsequently either enriched with a growth medium such as FPER2 or IMK, or fixed with glutaldehyde and observed using an Olympus CKX53 inverted microscope. The encountered cells were characterized using several identification keys, which we focus more on the diatoms and the dinoflagellates assemblages. So moving on to the results and discussions. With regards to the marine litter composition, the encountered plastic garbage comprised mainly of plastic bags and factory packagings, whereas the biological debris consists mainly of seaweed, seagrasses, and fallen leaves. Of all the garbage and debris collected, plastic bags were the dominant component in all of the sampling sites. And these findings are in agreement with previous studies by Cordova and Nurhati 2019, and also Hayati et al. 2020, regarding the dominating plastic garbage in the marine litter of the northern region of Jakarta. Subsequently, we detected a total of 48 microalgae taxa, of which 29 were diatoms and 19 were dinoflagellates. Of these, 18 taxa were successfully identified down to species level, and 34 taxa were frequently or commonly observed in the samples. The most diverse sites was TI03 with 19 taxa, including 5 potentially harmful species, while the least diverse site was TI06 with only 2 taxa. The 2 TI stations were located at the Tidung Island. And as expected, most of the identified taxa were part of the benthic assemblage. But interestingly, some planktonic taxa were also observed in the samples, and we thought that they were probably trapped in the plastic garbage during the dispersal voyage of the plastic materials, or they were unintentionally trapped during the sampling processes. This table shows the occurrence records of microalgae detected in each sampling station based on the type of samples, whether it was plastic or biological materials. The findings on microalgal cells, including harmful species in both the plastic and biological samples, indicated that plastic garbage can act as a replacement for the biological substrates for such taxa. This was particularly supported by species that were exclusively detected in the plastic garbage samples. Hence, we believe that these results provide a strong evidence for the potential dispersal of harmful microalgae via plastic garbage. With regards to the harmful microalgae, we detected 10 potentially harmful species in the collected garbage samples, of which 4 were diatoms and 6 were dinoflagellates, with 1 or more types of associated adverse effects. For example, 6 species of the 10 detected potentially harmful species were known to form massive blooms, of which 5 of them were able to produce toxin-causing seafood poisonings. One species in particular, with the Ceratoaulina, was reported to cause gill clogging in fish in other reports, while 2 others are associated with airborne toxin, especially Ostreopsis. In conclusion, our study was the first in Indonesia to provide a solid baseline information on the microalgae composition, especially harmful microalgae composition found in plastic garbage and biological debris. The presence of 10 potentially harmful species in the garbage samples is indicative that such material could serve as a hitchhiking substrate that could aid in their wider distribution in Indonesian seas, and therefore supporting our proposed hypothesis. However, we would like also to suggest that more sampling efforts need to be conducted involving other types of garbage, such as fabric or polystyrene, to understand whether different types of marine litters could also transport microalgae to different locations, especially in Indonesian seas. And it's also important that we can probably conduct laboratory experiments in the future studies to reveal the fate of microalgal cells during the transport when they are subjected to various environmental stressors, such as fluctuating salinity, temperature, UV radiations, winds, and currents. Furthermore, it is also important that we can also incorporate dispersal trajectory analysis of the marine garbage and debris in the Indonesian waters in order to assess the dispersal range and simulate the potential adverse impacts of the transported harmful microalgal species through the marine plastic garbage in Indonesian waters. Finally, we would like to thank Kurita Water and Environmental Foundations from Japan for providing the grant to conduct this research, and particularly and personally, I would like to thank all of you for listening to my talk. Thank you very much.
Okay, thank you very much, Pak Danang, for a very nice presentation about the potential disposal of harmful marine microalgae via plastic garbage. This is a very interesting topic, and we can discuss about this topic later after we finish for all the 6 presenters. Yeah. Okay, let's do the second presenters. Bu Hani Susanti will present the influence of sphagnum, feed extract on the biomass of novel algae, Ostracomonas triangulus, from peatland. For Bu Ani, please.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. Today presentation. My name is Hani Susanti. Would like to deliver our research report with entitled Influence Sphagnum Feed Extract on the Biomass of Novel Algae Ostracomonas triangulus, fulvocalis, chlorophysae from peatland. In this study, we use microalgae. Microalgae is microscopic algae. This photosynthetic microbe gets the attention for biomass production. However, only limited species are studied in detail, such as Botryophoca for biofuel production, Flora for single-cell production, and Chlamydomonas for many genetic engineering study. The source for algal biomass can be derived from various habitats, including peatland, or so-called in Indonesia, peatland is lahan gambut. Peatland is the unique habitat because of the low acidity and the high carbon content. Algae in peatland is interesting to be studied. Currently, our knowledge is limited, therefore, more information is necessary for sustainable development. Regarding the algal biomass, we have faced several challenges such as high production costs, contamination during cultivation, and low growth rate of selected strain. Now we introduce the peatwater to overcome these particular challenges. Peatwater contains organic material as a source of nutrient, and the acidity condition of peatwater may reduce the contamination agent. In addition, the biostimulant component of peatwater may support the algal growth. In the previous study, we published the novel species of algae with accession number NIES 4318. Is Chlamydomonas-like algae derived from acidic wetland or peatland. The identification of this new algae was reported by Susanti et al. In 1990 in Q1 journal Phytotaxa. The origin of habitat for NIES 4318 is in peatland, which were dominated by sphagnum plants. The aim of our research is to evaluate the utilization of peat extract water for biomass and oil production of novel algae Ostracomonas triangulus under acidic cultural conditions. For the method, we conducted the morphological study using microscope and continued by characterization of peatwater. In our study, we used Estonia peat soil contain dried sphagnum biomass. This kind of soil is quite cheap and easily to be found in the agricultural market. So then we analyze the biomass, we compare the biomass using commercial media treatment as AF6, which contain carbon up to 1% and the nitrogen contain up to 15%, while our pit water contain quite high amount of carbon up to 39%, but the nitrogen is low, only 1%. Then we analyze the lipid production using blood and dye method, followed by the fatty acid profiling using gas chromatography. The results show that for the growth of NIS 4318 is gradually increased during 15 days. The biomass production comparable between AF6 and pit water is not significantly different at all between AF6 and pit water. However, using AF6 treatment, the biomass production slightly higher than the using pit water. However, for the lipid production, the NIES produce the lipid higher using pit water treatment than using commercial media. We can explain here that the low concentration of nitrogen inside the pit water may enhance the lipid production. Sufficient nitrogen is widely known to enhance the lipid production in many algae. Here we provide the figure of oil droplet of NIES 4318 after staining using nyrate. The yellowish or golden color indicated the oil droplet inside the cell. We also provide the profile of fatty acid of NIES 4318. From this profile, we can summarize that the same algae may be a source of different fatty acids depending on the nutritive composition of culture media. In our study, the nutritive composition between AF6 different with using pit water. That's why the profile of fatty acids between AF6 media and pit water media is different. We also analyze the dominant fatty acid under pit water treatment. The results show that the production of palmitic and oleic acid is increased, while the linoleic and linolenic acid production is decreased. The total monounsaturated fatty acid as well as saturated fatty acid, the production is increased because of the pit water treatment, while the total polyunsaturated fatty acid is decreased. From our study, we can conclude that the novel algae Ostrophamonas triangulaculus and IES 4318 is able to grow well in acidic media as well as in the normal media. Sphagnum pit extract water is a promising source for the biomass and also for oil production. And we recommend for further research to enhance the algal biomass as well as oil productivity to be conducted. This is the end of my presentation. Thank you very much for your kindly attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bu Hani Susanti, for the very nice presentation about how to enhance the biomass and the oil production from the novel microalga, yeah, Ostrava monas trianguloculus. Yeah. Okay, so for all the participants, if you have any question or comment, you can write in the chat, and also you can deliver by open mic later after six presenter, and then we will continue for the third presentation by Bu Dini Akhnafani. Yeah, this is about the cloning and expression analysis of synthetic MHATase, yeah, gene under mutated well signal sequence in Escherichia coli BL21. Yeah, for Bu Dini, yeah, please.
Hello and good morning. My name is Dini Ahnafani. I am from National Research and Innovation Agency. Today in this opportunity, I will present our latest research by the title Cloning and Expression Analysis of Synthetic Metase Gene under Mutated PLB Signal Sequence in Escherichia coli BL21DE3. We will start from the background of our research. Like we all know, we use plastic in our daily activity, mainly for food container. Plastic mainly made from PET are lightweight, strong, clear, and low production costs. However, plastic degradation does not occur naturally, and now we commonly found it as microplastic in our land and ocean. Recently, novel PET degrading microorganism was found, namely Ideonella. It has the ability to degrade PET by two cascade system. They can produce PETase and metase enzyme, in which the PET will degrade into monohydroxyethyl terephthalate by PETase, and together they produce bis-hydroxyethyl terephthalate and terephthalic acid. The monohydroxyethyl terephthalate was then further converted into terephthalic acid and ethylene glycol by metase enzyme. Now we will focus on the metase enzyme, as it is predicted as a membrane-bound enzyme, and it is a challenge itself to produce the soluble and active metase. Other works done in 2021 successfully expressed extracellular adenylosaccharins PETase by fusing the PETase gene with mutated PLB signal sequence, in which the 20th amino acid alanine was changed into threonine. Based on this data, we tried to express the extracellular metase enzyme by fusing the metase gene with mutated PLB signal sequence and analyze the protein expression by Western blot and esterase activity. This is the method we use in our research. For the recombinant metase gene, we first translated and codon-optimized metase amino acid and fused it with PLB signal sequence, in which the 20th amino acid alanine was changed into threonine. The gene was then synthesized by GenScript. We cloned the gene into PET22B and transformed into E. coli DH5 alpha. The verification was done by NDE1 and XHO1 restriction enzyme. The protein expression was done by growing the recombinant bacteria in Luria bertani media using the ampicillin as a selective antibiotics marker. The IPTG induction was done after the optical density reached 0.8 and the induction performed for 5 hours at 37 degrees Celsius. The induced culture was then centrifuged to separate the cell-free media and the cell pellet. The cell-free media here, we used it for the extracellular protein fraction sample. As for the cell pellet, we use it for the extraction of the protein into the periplasmic protein fraction and intracellular protein fraction. For the periplasmic protein fraction, we use osmotic shock extraction protocol with osmotic shock buffer containing 20% of sucrose. As for the intracellular protein fraction, we use sonication to lyse the cell pellet using the lysis buffer and obtain the soluble fraction. As we use E. coli expression system, they might be found our target protein as insoluble fraction or inclusion bodies. So we use the unfolding method here, containing urea, to unfold the protein aggregates. And further, we refold the protein using the refolding buffer containing N-lauryl sarcosyl. After all those steps, we have 4 different protein fractions here, and we purified those proteins using nickel resin system. And the purified protein fractions were analyzed by protein content analysis using Bradford method, esterase activity using polynitrophenyl acetate hydrolysis, SDSPH, and Western blot. This is the results of our experiment. For the recombinant metase gene, this is the plasmid construct we use for the metase expression. We can see here that the metase gene is cloned in between XHO1 and NDE1 restriction site. The digestion using those 2 enzymes resulting 2 gene with 1.8 kilobase for the inserted DNA and 5 kilobase for the vector. With this result, we can conclude that the metase gene was successfully cloned into NDE1 and XHO1 restriction site of the PET22B and successfully transformed into E. coli BL21DE3. In the protein recovery, as I mentioned before, actually we have 4 different protein fractions, but 2 of them, periplasmic and extracellular protein, did not show any protein band in the expected size for about 62 kilodalton. Even after concentrating the lysate using vivaspin, we still can't see the protein. So we conclude that the metase enzyme was not produced as extracellular protein. Here we have the intracellular protein fraction. Both are cell lysate, that is the soluble fraction, and the solubilized fraction that comes from the solubilized inclusion bodies. Qualitatively, we can see the difference in the band thickness of the SDH-based electrophoregram. Here we can see that the protein at the 55 to 70 kilodalton here are thicker than the cell lysate. After the purification process, we obtained 1.789 micrograms of purified metase, and it is about 60% of the total metase enzyme expressed in E. coli. This is the SDH-based electrophoregram of the metase purification. We can see clearly in the solubilized fraction here that the protein band was distinctively observed in solubilized fraction with less contamination, while no significant band at the expected size were observed in the soluble fraction. Here we also have the refolded metase, and it still can be clearly observed at the 62 kilodalton. And this is the Western blot analysis using the anti-HISTEC monoclonal antibodies. The left one is from the soluble fraction and the right one is from the insoluble fraction. Both fractions shows a clear band signal at around 55 to 70 kilodalton, but we can see that the protein band was expressed at a higher level in the insoluble fraction here. Even though many signal bands here detected in the insoluble fraction, they might be due to the histidine-rich amino acid produced by the E. coli itself. For the esterase activity, the recombinant metase here yielded 0.4 unit per milliliter of esterase activity, and it shows that the refolded metase was functionally active. So in conclusion of our research, first, the recombinant plasmid PET22B containing metase gene with mutated signal sequence PELB was successfully cloned and transformed into E. coli BL21DE3. The recombinant metase was suggested to be majority expressed as inclusion bodies with 1.5 times higher than soluble fraction. The solubilization and refolding steps of the metase inclusion bodies was successfully performed and yielded active metase with 0.4 unit per milliliter of esterase activity. And these are some references for our experiment, and I would love to hear some feedback from you. Thank you for your attention.
Okay, thank you, Bu Nur Absari Aji, for the very nice presentation about the cloning and expression gene of mitase, yeah, gene under E. coli. And yeah, again, I remember for all the audience, if you have any question, you can write in the chat and we can discuss the question and answer in the next later after 6 presenters present their progress, their presentation. Okay, we continue to the to the 5th presenter, Bu Uri. No, sorry. Now is Bu Nur Absari Aji, yeah. It is the same, almost same topic with the Bu Dini Akhnafani, yeah. The title is cloning and expression of polyethylene terephthalate hydrolase, yeah, PTase, yeah, a gene under mutated 12 signal peptide in Escherichia coli, yeah. Bu Nur, please.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Thank you for the opportunity. Today, I would like to present our group project on cloning and expression of polyethylene terephthalate hydrolase PTase gene under mutated PLP signal peptide in E. coli BL21 DE3. On this occasion, I'm Maria Ulfa from Center for Applied Microbiology, will represent my group. Talking about PT or polyethylene terephthalate, we're talking about plastic. It is simple thing and very close to our daily life. We meet and use them every day. PET is thermoplastic prepared by polymerization of terephthalic acid and ethylene glycol through the formation of ester linkage. And PET commonly used as package for single-use consumer goods. But this surely contributes to a rise in the volume of PET waste and become a major environmental problems. So then, the increasing amount of PET certainly requires processing to be able to degrade them. And in 2016, Yoshida et al. Has discovered a bacterium that can degrade and assimilate polyethylene terephthalate. And the first PTS was discovered from Ideonella sakiensis bacteria, which found from sludge sample of PET bottle recycling site in Japan. So PTS, it's an enzyme that can ability to degrade PT polymer into the monomeric mono-2-hydroxyatyl terephthalate or MHAT. And this is MHAT can decompose MHAT into the ethyl glycolone and terephthalic acid. So in this study, we attempt to combine the engineered ISPT to improve the thermal stability and with the mutated signal peptide PLB from PET22 feed cloning expression factor. By this construction, we hope that the recombinant ISPT will be more stable and secretable into the medium directly. So now to enhance the DNA performance, we took a synthetic approach for cost-effective alternative. So we employ synthetic DNA to develop ISPT biocatalyst. Here, we highlight the process of the optimizing expression and recovery method of the recombinant ISPT in the E. coli BL21 DE3. In the beginning, the recombinant ISPT was propagated into the E. coli DH5 alpha, and then the screen into the plate, LB plate supplemented with ampicillin, and then transferment extracted by alkaline method, and analysis was performed with NDA and XAO extraction enzyme. For the protein expression, the recombinant E. coli harboring mutated ISPT was cultured in the algae broth containing ampicillin antibiotic, and 1% of the overnight cultures inoculated in the medium and incubated at 37 degrees until the OD reached 0.6 to 0.8. And after that, culture was induced by 1 millimolar IPTG, and recombinant protein was conducted in 37 degrees Celsius for 5 hours. And then the cell culture were harvested by centrifugation. The supernatant was stored at 4 degrees, while the pellets stored at -80 for further analysis. And to confirm the presence of expressed recombinant ISPTS, the extracellular fraction, peripragmatic fraction, and intracellular fraction were extracted using different methods. So as we can see in the picture that line 1 and line 2 are extracellular fractions. This obtained from culture supernatant by centrifugation and was concentrated by using Vivaspin with 10 kilodalton cutoff. The line 1 is control and induced protein. This is 1,2 is supernatant with IPTD injection. And meanwhile, the line 3 and 4 represent the periplasmic fraction. Is recovered by using the osmotic shock method. So the line 3 is control of induced protein, and the line 4 is represent the periplasmic fraction with IPTG induction. We can see the difference here. And this is the intracellular fraction, as we can observe that an increased intensity of protein was observed. But intracellular ISPTS fraction, especially in this line 6 compared to uninduced line 5, and this is the last line 7. It is also an insoluble fraction which further extracted by using the unfolding buffer. So based on this observation, it may assume that the recombinant ISPTS is in the cellular and periplasmic fraction, but it mostly observed in insoluble fraction. And then to confirm the specific protein target, Western blot analysis was conducted. So as we can see in this picture, this SDS is correspond to the Western blot analysis. In the intracellular fraction, the band target was well observed, and especially here in the line 2. This is the uninduced protein correspond to the line 1. And so from this result, we confirm that protein bands that appear with high intensity with the SDS page analysis under 35 kilodalton. And this is related to the weight calculation, which around about 30.7 kilodalton. So as this conclusion, we assume that the quadrupole mutation variant of ISPTS was successful in the mutated signal peptide PLB was successfully constructed in PET22, and the recombinant PTS was also successfully expressed on the E. coli BL21DE3 and induced by 1 millimolar IPTG at 37 degrees for 5 hours. But however, the extracellular and periplasmic fraction had lower intensity than the intracellular fraction. So this raises hypothesis that the secretion process might not be optimal. So further optimization and investigation needs to be done. Okay, thank you very much for your kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Bu Nur, for the presentation. And okay, we will next for the fifth presenter, Bu Wuri Handayani, with the title Attack of Suit-Sucking Pests on Gerunggang in Peatland Rehabilitation. So Bu Wuri, please.
Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. I will present my paper entitled Attack of Shoot-Sucking Pest on Gerunggang, Cratoxylum arborescens fall bloomer in peatland rehabilitation. The research team are Wuryandayani, it's me, Antiwinara Suhartono, and Endah Suhendah. Background of the research. As we know, Indonesia is one of the countries with a large peatland area. It is the fourth largest after Canada, Russia, and the United States. Peatlands are critical because they contain the most carbon in the world, serve as life support system, water and food sources, support biodiversity, and regulate global climate. However, peatland management is very complicated due to its fragile nature. Peatland in Indonesia continue to suffer from land degradation. Population growth encourages the conversion of peatland into agricultural land, plantations, and settlements. Slash and burn sometimes still used to clear peatlands as it saves money and energy. However, the ability of peat ecosystems to recover naturally is very slow. So the function of peat must be restored through revegetation with native plants. Revegetation with native plants will survive and grow better, but it should also be able to become a source of income for the local community. Gerunggang, Cratoxylum arborescens, is one of the pioneer species that is widely used for revegetation. Gerunggang has promising benefits and economic value, although in Indonesia the use of gerunggang trees is still limited to timber. Unfortunately, complete information on gerunggang cultivation techniques is still very limited because interest in revegetation of this plant is relatively new. Besides, gerunggang is a lesser-known tree species. The available information is limited to only the early stages of cultivation, such as nurseries or early growth of revegetation at the field level. Meanwhile, information about pests and diseases of gerunggang plants is rarely reported. So, this study aims to measure the intensity of gerunggang pest attacks on the peatland rehabilitation in Kubu Raya, West Kalimantan. The research was carried out on a demonstration plot for peatland rehabilitation in October 2018 in Rasawijaya Second Village, Rasawijaya District, Kubu Raya Regency, West Kalimantan. The pit depth is less than 3 meters. Pest disturbances were observed on gerunggang vegetation in the peatland rehabilitation plot with an agroforestry pattern, combining some species of native peat wood with fruit trees, namely jelutung, ulai, pinang, and jengkol. The spacing is 4 meters by 3 meters. This study used a 50% sample size and obtained 49 gerunggang plants. The number of plants, number of shoots, number of pest population per plant, and number of pest population and number of shoots attacked by pests were all manually calculated using a hand counter. Pest identification and attack symptoms are carried out directly on the research field. The attack intensity formula is based on the 1963 Untersteinhofer criteria. The formulas we applied are to get or to know attack incidence and attack intensity, as shown on slide. The variables needed in the attack incident formula are the number of plants attacked by pest and the total number of plants. Meanwhile, variables required in the attack intensity formula are the number of plants observed in each of the same attack categories, scale value of each attack category, scale value of the highest attack category, the number of all plants. Table 1 is used to determine the level of damage. The criteria for the level of damage are explained in terms of how many leaf shoots are attacked by pests. The level of damage is expressed narratively and also with numbers to determine the category of damage. Results and discussion. Gerunggang plants in the demonstration plot indicated being attacked by shoot-sucking pest and identified as Myctis sp. You can see here the imago of Myctis in figure 1. According to the literature review, there are plant species that have been reported to be attacked by Myctis sp, such as Mangifera indica, Lagerstroemia speciosa, Kaya inforensis. And this study is the first to report that Myctis sp has also attacked Gerunggang species, particularly in the research site. Symptoms of Myctis attack are indicated by the presence of liquid suction spots on the leaves and on the leaf shoots, as shown in figure 2. Myctis sp has sucked the sap fluid required for plant growth, causing the growth process of leaf shoots to be disrupted and the plant to be stunted. You can see in the figure 3. Attack incident and attack intensity. Attack incident Myctis sp is 100%. It means all plant samples, all Gerunggang samples are attacked by Myctis sp pest. The level of damage to individual plants was mostly in the moderate category, which indicates shoot damage more than 25% until 50%. There are 29 plants in the moderate category. The attack intensity or the severity of the attack on the Gerunggang sample reached 50.56%, are classified as a heavy pest attack. In this slide, we can see the difference between healthy Gerunggang plants and Gerunggang that heavily attacked by Myctis sp pest. As I mentioned earlier, Myctis sp attack caused shoot death, didn't cause plant death, but cause shoot death and disrupt plant growth processes, and then the plant becomes to be stunted. This is, of course, can reduce the production of Gerunggang. Conclusion: insect pest species Myctis sp had attacked all Gerunggang, or the pest attack incidence reached 100%. The attack intensity reached 50.56%, or was classified as a heavy pest attack. Myctis sp pest attack does not cause plant death, but their abundance can interfere with plant growth and decrease the potential of Gerunggang production. Thank you for listening my presentation. I hope what I have shared will be beneficial for all of us.
Okay, thank you very much, Bu Handayani, for a very nice presentation about the attacking of suit-sucking pests, on Gaunggang. This is very interesting study. Okay, we continue to the six presenters, Bu Triwira Yuwati, with the title Potential Plant Species for Sustainable Livelihood in Peatland. Bu Triwira, please.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. My name is Triwira Yuwati. On behalf of my colleague, Doni Rahmanadi, Purwanto Budi Santosa, Dian Pratiwi, and Pamungkas Buana Putra, we are from Research Center for Ecology and Ethnobiology, National Research and Innovation Agency, BRIN, who would like to present our paper entitled Potential Plant Species for Sustainable Livelihood in Peatland. Lesson learned from demo plot establishment in Central Kalimantan. So this is the table of contents of my presentation. First, about introduction, and then followed by land-based activities of peatland utilization in Central Kalimantan, and then demo plot establishment and the species planted, potential plant species and routing system, and I will end up with conclusion. So according to ANDA 2021, Indonesia's peatland covers an area of approximately 13 to 14 million hectares. And tropical peatland has important functions, as the preservation of biodiversity, the quantity and quality of water, the prevention of flooding and soil intrusion, erosion and sedimentation, and the provision of livelihoods for the community. But drainage, logging, and fire has caused peatland degradation in Indonesia. And to restore the Indonesia's tropical peatland, the government of Indonesia has established the Peatland Restoration Agency, or Badan Restorasi Gambut, in early 2016. And after the restoration efforts, there are still problems remains, which is resolving issues of recurrent fires that inhibit the revegetation, and also how to provide stable livelihood for the community living around or in the peatland. So due to several obstacles such as flooding, soil acidity, low soil fertility, fire, and a limited selection of plant species, the utilization of peatland in many Indonesian villages has become a concern. Due to the shifting choices for land-based livelihood, it is vital to plan ahead during the transition from drain pit to fully rewetted pit. To create a strategic model for the transition from the low water table to fully rewetted pit, adopting agroforestry to manage income and cash flows ranging from agricultural species to peat swamp forest species, many demonstration plots in Central Kalimantan have been established. So the aim of our paper is to determine the potential plant species for sustainable livelihood in peatland. So as you can see here, the above picture is actually the peatland in Central Kalimantan. So peat hydrological unit, or KHG, is a peat ecosystem located between two rivers, between the river and the sea, and or in the swamp. So the type of land located on the banks of the river is generally land with mineral soil or alluvial, up to the natural embankment, which is the border with the type of land with peatland. Most of the villagers in this typology who have land-based livelihoods are in the mineral land type, in the form of rattan plantations, rubber plantations, and also paddy fields. Well, the peatland that is overgrown with forest is used by the community to find timber, forest products, or non-timber forest product, and also hunting. So a small part of the community works on peatland by planting paddy fields, but that is limited to only in thin pit, that is around 50 centimeters to 200 centimeters. And for the case of the Central Kalimantan, there are many villages that are located in the protected area and practically try to use the land as a source of livelihood. So the culture of using mineral land from the village that used to be located on the banks of the river was tried to be applied in peatlands, such as making rubber gardens, fruit gardens, pineapples, or in the form of using yards with the mixed gardens and so on. And cultivation of plants that come from dry land in peatland is becoming more and more widely practiced along with the progress of the transmigration program, so that there are many variations in the utilization of peatland. So as you can see from the above pictures, this is what happened in Central Kalimantan. So by trying to plant crops on deep pit, you need to drain the pit. And by the pit drainations, by establishing canals, then it is also becoming, the pit also becoming prone to fire. And with fire, of course, it will increase carbon emissions and also habitat loss. But the most important thing is also impact on the livelihood of the local people. And the pictures below is the pit, what we will achieve with the peatland restoration. As you can see that the crops only planted on the thin pit, and in the pit dome or deep pit is only for perennial local peatland species and also non-timber forest product. So commodities developed in peatland should be suitable for wet peatland conditions and can also support peatland improvement activities. So the use of commodities that come from dry land turn out to have various disadvantages, such as increased land emissions and drying of peatland due to the canal constructions, as I have mentioned before, and a lot of information about the failure of agricultural businesses that occur due to the differences in the nature of peatland and mineral land. And these are the demo plots that has been established by the FORDIA, Forestry Research and Development Agency, Banjarbaru, ranging from 2003 until 2020, with also various commodity or species that has been planted, and also in various locations, which is it is in private land and also in state-owned land in Kahadika Tumbanusa, also ranging from the wide of the plot area, ranging from 1 hectare until 50 hectares. And we then group or analyze based on perennial or non-perennial plant species from the demo plot establishment. And these are the perennial species, which is Alsonia penematopora, Dira polypila, Sorea belangeran, Combretocarpus rotundatus, Conistrus balcanus, Cartoxylon glaucum, Notafibi coreacea, Nephelim lapasium, Areca catechu, and also non-perennial such as Covea librica, Ananas comosus, Colocasia esculenta, Solanum melongena, Solanum lasiocarpum, Capsicum frutescens, and Vigna unguiculata, with relative growth rate for height, survival rate, and also months after planting. And understanding the character of each species in the suitability growing niches in the specific rhizospheres and pedosphere is a basic knowledge of revegetation techniques for rehabilitation and restoration program. So Sorea belangeran is able to form buttress root in adapting the inundation. Dira polypila also has a unique characteristic in the form of presence of knee-shaped pneumatophore roots. And also Alsonia pneumatopora simultaneously form buttress root and knee-shaped respiratory root system that grows above the surface of the water. So these three species has adapted themselves in the rewetted peatland. And the findings of this natural phenomenon can be adapted for peatland rehabilitation techniques through the manufacture of mounding or mounds for cultivation and planting of introduced seeds. And the heights of the mound is adjusted to the water level during the dry season and also the rainy season. And as for non-perennial species, pineapple is chosen as a non-perennial introduced species in peatland cultivation because it is not only able to adapt to various soils, including peat soil with low pH, but is also able to form advantageous roots to support its life. So in conclusion, there are three perennial local peatland species, Sorea belangeran, Dira polypila, and Alsonia pneumatopora, and one non-perennial species, Ananas comosus, showed good performance and potential to be developed for sustainable livelihood in rewetted peatland. I will end my presentation. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Bu Yuwati, for very nice presentation, and all the participants. Now we will have a question and answer discussion for 20 minutes. For all the audience, if you have any question or comment, please open your mic, and should you should have mentioned to address your questions for the presenters, please.
Can I ask the question?
Okay, please, Pak Panji.
Yeah. My name is Panji. I am from Research Center for Environmental and Clean Technology, BRIN. I have a question for Pak Danang. Pak Danang, I am particularly interested with this topic, and something that I took by heart because I did my PhD in microbial invasion. So I am very interested in, in terms of dispersal of this microalgae. Do you think that the environmental parameters would affect the dispersal of the microalgae, or is it more stochastic or random in a way? That's my first question. The second question is, I saw that you used the microscope to characterize the microalgae communities. Would you think that using metagenomics or amplicon sequencing, such as the 18S amplicon sequencing, would characterize the microalgae better instead of the microscope technique? Thank you very much.
Okay, thank you very much for Panji. And okay, please wait for the answer, ya Pak Danang ya. We will invite more one more question. If you have any question, please.
Thank you very much.
Okay.
Sorry.
Bu Sri, yeah, please.
Yeah, I have a question for Ibu Trivira. Yeah, is it?
Yeah.
Sorry. It's a very nice presentation to know that actually we have an options to support livelihood option for the people around the peatland. So based on your research, is it those three species that you recommend in this demo plot is able to support people livelihoods nearby the peatland area, and then how to engage community around this area to also planting these species? Thank you.
Okay, thank you. We have and one is for Bu Tri. Please, Pak Danang and Bu Tri. Maybe first for Pak Danang. So to have you, please answer this question.
Okay, can you hear me?
Yes, yes.
Okay, thank you very much, Pak Dede, and thank you very much, Pak Panji, for your questions. The first question was about the effects on environmental factors, whether it can affect the dispersal potential of harmful algae through plastic garbage. My direct answer actually is yes, it can affect, because once the microalgae or harmful microalgae are attached to the plastic debris or garbage, it will then susceptible to various environmental effects such as winds, currents, fluctuating temperatures, salinity, and so on. And this is what we suggested actually in our manuscript, that in the future we should conduct the tests to determine the fate of those harmful microalgae or microalgae in general that are attached to the plastic garbage. So my direct answer to that is yes, it will certainly affect to certain points, or depending on the species and so on. But my direct answer is that. And the second question is, at the moment I'm using a microscope because our research was actually a preliminary study to determine whether harmful algae are attached to the plastic, and the easiest way to determine that is by looking through the the microscope and look directly at the cells. But I agree with you that another techniques to determine the diversity or composition of harmful microalgae using molecular techniques is much better. In other way, I'm actually a taxonomist, so I'm very much confident with the microscopical observation and identification techniques, and I think that is much more faster than using molecular. But we have also collected samples for ED and metabarcodings actually from marine plastic garbage. So we are looking forward to receive, hopefully to receive another grants to determine the ED and metabarcodings of the samples from the plastic garbage. So, well, hopefully, if you are interested, then shoot me an email or something like that. We can discuss, and probably we can collaborate or something with that if you are interested, Papanji. I think, I hope that's answering your questions. Thank you very much.
Thank you very much. Yes. Can I ask the follow-up question, or should I just finish it here, Pak Moderator? Hello, Pak Dr. Heru.
Are you there?
Can I ask follow-up question, or should we move to another question?
I think Pak Didi is out, but maybe you can shoot out a short questions directly so I can answer.
Okay, yes. Short question. How can we limit the dispersal then in this case? Do we have to reduce the plastic garbage, or there would, you know, do you think there are more things that we need to control in order to limit the dispersal of the dangerous microalgae?
Well, there are many ways on how harmful microalgae can be dispersed in the oceans. Well, plastic garbage is one thing. Naturally, they are dispersed through the floating biological garbage, such as leaves, parts of seaweeds or seagrasses, and so on and so on. But we have to understand that those that are dispersed through such materials or floating materials such as plastics, biological garbage, and so on are epiphytic species that attach to them. So, but we have to understand that harmful microalgae also consists of planktonic species that are not attached to that. Another way of how harmful algae can be dispersed is through the currents. So it happens naturally. But our idea of this paper was to highlight the detrimental impacts of plastics in the environment. So we would like to emphasize that we have to take a great efforts to limit the plastic pollutions, because the plastic pollutions can disperse evidently the harmful algae. So that's actually is the main idea why our study was conducted. Yeah, you can shoot me a chat or something like that, or emails, and we can discuss further if you are interested. Thank you very much, Panji.
Yeah, thank you very much, Pak.
Okay, thank you very much, Pak Dana, for a very nice answer. I also interesting about this issue. Yeah, continuing with this question also, Pak Dana, can I have a question for you?
Yeah, sure.
Okay, so Dana, is a different kind of plastic garbage will have a different kind of harmful algae presence in the plastic garbage? This is the first question. And why? I mean, from where the microalgae present in the plastic garbage? Yeah.
Okay.
This is my question.
So, okay, thank you very much for your question. So the first is whether different types of garbage or plastic garbage will harbor different kinds of harmful algae. And in our study, we haven't determined that, because we saw that both type of species can be found in both types of garbage, either plastics and biological samples. So at this moment, we cannot answer whether certain types of harmful algae, preferring certain types of garbage or materials. But it will be very interesting to have a statistical analysis and more sampling efforts on that to determine whether certain species or genus or groups of harmful algae can or prefer certain types of materials. That's my first answer for your first questions. The second is regarding why harmful algae can attach to plastic. Is that your question?
Yes.
Okay, so some microalgae or harmful algae in particular are epiphytics. So they can attach to a substrate like rocks, the surface of leaves, or sea grasses, and so on. So naturally they can attach by certain morphological features or cell features that they have. So how they can attach to the plastic is you know, I explained in my presentations, when somebody throw out the garbage to the oceans and the sink, some species that are epiphytics or benthics in nature, they move or they move to that plastic garbage and they attach themselves. So I'm not sure how the mechanisms or whether there are a certain preference whether they attach to that at the moment, but our study indicates that yes, we found species that are attached to that. Even under microscope, there are species that need to be strapped strongly so that we can obtain the cells. Will that answer your questions? I hope so.
Yeah. Okay. Very interesting. Thank you.
Thank you
very much again, Pak Dana. Okay, for the next answer, please, Bu Tri.
Thank you, Mr. Moderator. Thank you, Ibu Sri Lestari, for your questions. Well, for the first question about whether the species can support the livelihood, yes, they can, because not only, well, if you see at the timber product, it's very long time, but actually this species is also a non-timber forest species, for example, like latex for Jelutung rawa, and you can also harvest the latex. And also the most important thing is the seeds. So these three species are very high producing seeds for the peatland species. And as you know that you said your second question is how to engage the community to also plant the species. Well, actually, not only the socialization, but also people will look at examples. So as we have the demo plot here in Central Kalimantan, especially in the Tumbang Nusa village, is the center for the nursery, community nursery. So with this nursery business, it's growing a lot in Tumbang Nusa. Even for Jelutung rawa, once we have calculated for 1 hectare, the farmer can obtain 18 million rupiah in one harvesting time. So it's very how to say it? It's very promising for the livelihood. And I think that's answering your question, Ibu Liz. Thank you.
Okay, thank you, Bu Tri. Yeah, I invite more questions for the audience. We still have 7 minutes again. Please, is there any question? So I have a question for Bu Wui. So Bu Wui, I saw in your presentation that Mystic SP species attack on Gunggang is categorized as a heavy pest attack. So what is the implication of this attack, and also how to, I don't know, how to control this Mystic species from attacking on Gunggang?
Can you repeat your question?
So in your presentation, yeah, Mystic
Yeah.
species attack on Gunggang and it is categorized as a heavy pest attack, right?
Yeah.
And so, what is the implication of this attack? What is the implication? I mean, is it? I cannot describe about the dangers of this attack or something like that. Can you describe that and how to control this attacking from the mystic species on Gaunggang?
Yeah. The implication, the plant cannot grow perfectly. There will be plants become stunted, so it will be decrease the potential. Yeah, the potential of the timber, the Gurunggang timber, and then how to control, how to, I'm sorry.
How to control the mystic attack? For example, by using the pesticide or?
I'm sorry. I think
maybe by using the. Yeah, please, Bu.
So, I'm sorry.
Some pesticide can control the mystic to attack Gurunggang. Yeah, I don't know, but anyway, thank you, Bu Wei. Is it okay? Yeah, I invite again for another question from the audience, please. A comment from the audience. Okay, so I have a question for Bu Dini and Bu Nurul, yeah, or Bu.
Can I answer your question?
I show active.
How? Tadi. Pak Danang?
Sorry, I think Dr. Dede get a problem in the signal. Please,
Pak Dede, sorry. I'm sorry,
you can answer the question of
Okay. Yeah, we can control the pest if the population, yeah, can also be manual capture or by spraying chemical pesticides. That's my answer. Yeah. The type of chemical pesticides based on acipate or dimethoate. That's my answer. Okay.
Okay, so to control the insect, you suggest to use the pesticide. Okay, thank you. So, if there are any question, I think Dr. Dede is already here.
Okay, thank you, Pastor Imam. Very sorry because I'm on the mountain area now. So, yeah, is there any question or comment again? Okay, I think I have so many questions, but time is over, and so can we finish this session, Mas Imam?
Yes.
Yeah.
So,
Okay. Yeah. Okay. Thank you very much again.
Thank you, Dr. Dede, and all participants. You can return to the main.
Yeah. Thank you very much for all the presenters and audience. Thank you.
Thank you very much. Pak Imam, punten. Shall we
Yes,
leave
please.
the room or we stay this room?
Yes, you can leave the room, move to the main room.
The main room. Okay, thank you very much.
Recording in progress.
Distinguished guests, ladies and gentlemen, welcome back to the main room. How was the parallel sessions, Bapak, Ibu, and also ladies and gentlemen? We hope it was very nice sessions. And now we will have a break for approximately 1 hour and 5 minutes. Please kindly remain in the Zoom meeting or back to the Zoom meeting at 1:30 p.m. Jakarta time. Once again, we will have a break for approximately 1 hour and 5 minutes. Please kindly back to the Zoom meeting or remain at the Zoom meeting. And we will start the next agenda at 1:30 p.m. Jakarta time. Thank you.
you.